I'm going to talk on the clonal origin and evolution of multisystem histiocytosis in adults, and I'd like to thank the organizers for the opportunity to present this data. So this is a story about family trees, and you can see on the left here a very famous European family tree. This is the Spanish royal family, and human family trees like this are often directly observed with known relationships between the grandfathers and the children and the grandchildren. I know this because it's reported to me by the Daily Express, which is a very reliable English newspaper. And family trees, or as we know them, phylogenies, in biology are very powerful tools for analysis of evolution, as you can see in this diagram, but also of epidemics, as in the COVID pandemic, and in cancer. Now, the reason they're powerful is you can, starting from the grandchildren or the descendants, which is where you have the data, you can infer back the relationship between entities within the family tree, so who is the closest. And you can also, if you have some standardization of the rate of evolution or the rate of mutation, you can infer a common ancestor and the sequence of events and how far back events occurred. So if you translate this to a single organism, we're all ultimately derived from one cell. So in principle, you could take a number of single cells from one person and figure out the relationship between the single cells and how they originated. So for example, these two cells share a red and a blue mutation. Sorry, this is by sequencing the genes of the cells. So you can infer that these were both derived from a common ancestor, whereas this third cell has unrelated mutations, so must arise further back in development. So you observe these cells and you infer these relationships. And this is often expressed in a more formal way as a form of phylogeny here, where each leg is is marked by a mutation, and these are the branch points where inferred relationships where cells group and ultimately go back to a common ancestor. So this is where the data comes, and this is calculated back from comparing the similarity from one cell to the other. So you can do this in a human. You can start with clones of blood cells, each one sequence the genome and figure out the evolution of these cells from a single fertilized egg. This is a developmental study that allows relationships and timing between embryonic tissues as they develop. So it's a very powerful method. It's also been applied to cancer in the example here of chronic myeloid leukemia. So down this y-axis, you have conception. At this point, you have the development of normal clones of blood cells. So each one of these is a whole genome sequence. And you can see at some point, roughly 5 years or a couple of hundred mutations before the presentation of the leukemia, there was a Philadelphia chromosome event here resulting in a very rapid clonal expansion causing chronic myeloid leukemia. So you can pinpoint the time of this mutation, you can measure the selective advantage for this clone, and you can see exactly how the disease evolves in great detail. So we were keen to apply this kind of analysis to histiocytosis in order to address these kind of questions. So how far back did it start? What is the sequence of events if there are multiple events? And how do these mutations interact and evolve? And this potentially might be useful for surveillance or early diagnosis of patients, and it might even offer some new therapeutic approaches. For example, if we see specific events occurring early on that have clonal advantages, there may be some way to modify these pharmacologically. And there's a recent example of this in DNMT3A clonal hematopoiesis, which can be modified by metformin, which reduces the advantage of the clone. So how is this done? You will start with a patient, sample some blood or bone marrow, and grow a number of single-cell colonies. So this step is important as a technical step because you can't sequence the whole genome of a single cell without introducing a lot of error if you do a whole genome amplification. You have to rely on the expansion of the cells by cell division, which is a nice error-checked process in order to gain enough DNA to sequence multiple genomes. And then you can construct this back into a phylogeny and ask the questions. So this is our experiment. This is the first analysis of the data, so we still have to do some more work on this. And we started with 7 patients with Erdheim-Chester disease, a range of ages, a range of BRAF frequency. This is in the mononuclear cells of the blood. And from this, it's my colleague Paul Milne. He grew over 1,000 colonies from these 7 patients. And this shows the sort of technical difficulty of it. From 1,000 colonies, we had 14 with BRAF mutations. So these are all BRAF mutated patients. And we wanted to identify BRAF positive colonies so that we could make sure they were represented in the tree. And these have gone forward for sequencing, and I'm going to show you the preliminary analysis of these 2 phylogenetic trees. So they all have BRAF, and you can see that they were quite heavily co-mutated with other events, other clonal hematopoiesis mutations. But we didn't select them on this basis. We essentially selected them on the ability to grow colonies and to select BRAF positive clones in the experiment. Just to give you a brief overview of the patients, there were 2 patients, a woman aged 59 and a man aged 73. They both presented with relatively indolent findings of skin rash. This patient had a mild leukocytosis at diagnosis. The skin showed Langerhans Cell Histiocytosis. She then unfortunately suffered a stroke, which left her quite disabled and led to CTs and imaging, which revealed Erdheim-Chester disease in multiple sites, confirmed on biopsy of the bone and the perirenal tissue. The male patient presented with, had a 10-year sort of pre-drome of mild neutropenia. He had a bone marrow at this point, which was reported as slightly reactive with no dysplasia. He was presumed to have an autoimmune neutropenia, but he subsequently developed constitutional symptoms with a rash. This was confirmed as Langerhans Cell Histiocytosis and also found in multiple sites, including the lymph nodes. He then had PET scans showed several sites of Erdheim-Chester disease involvement of the aorta, the bones, and bone biopsy confirmed Erdheim-Chester. And this is what the pathology looked like. I won't go through this in detail, but you'll see convincing evidence of Langerhans cell in both patients in various sites, and also Erdheim-Chester features in the perirenal and the bone biopsy of both patients. So they essentially had a high-risk mixed histiocytic picture. So this is the first patient. So this is our view of the clonal evolution of histiocytosis in this 59-year-old woman. And again, the y-axis shows the development of her mutational status from the point of conception. This is inferred at the moment because we have to sequence some more so that we can completely calibrate this axis back to the start. But essentially, we can see that the first thing that happened, she acquired these 2 TET2 mutations. And we know from adjusting the known rate of mutation, calibrating this to years of life, so this is early 20s, that she'd acquired a clone of TET2, biallelic TET2 mutation by her early 20s. And then something very remarkable happened, which may be linked to the high-risk presentation of her disease, ultimately, that she, within the space of 5 years, so that's from this point here to this point here, this is roughly a 5-year of her lifespan, she developed 3 parallel MAP kinase mutations, an NRAS, a BRAF V600E, and a KRAS mutation. Then there was another almost 15 years or so before she presented with any disease. So this tells us that was a very complex genetic landscape, and it was evolving for several decades before she presented with any symptoms. So remarkably, the second patient had a very similar structure to the clone of their malignancy. So again, a double TET2 mutation, which had established itself probably as a small clone in the bone marrow by the age of 40 or so. Remember, this patient was older when they presented. And following that, an interval of a couple of decades or so before he then developed, again, a triple KRAS, BRAF, and NRAS mutation, all in separate clones within 7 years or so of life. But then another period of latency, another 10 years or so before he presented with histiocytosis. At this point, he may have had a mild evidence of, this is when he had neutropenia, so wasn't completely silent, but he continued to work. I think he was a dentist. He worked beyond the normal age of retirement. He was basically fit and well while all this was happening. One of the things that we can take from this data is the molecular signatures, so we can look at the potential drivers of these mutations at the biochemical level, and the red and the blue here simply indicate the normal time-dependent, proliferation-dependent base changes. This is the SBS1 signature, for example, is a deamination of methyl-cytosine. So this is commonly seen in all other kinds of neoplastic blood disorders. So there was no extra mutational event, no other exogenous mutagen as far as we know. And so this is, in summary, this phylogenetic analysis. I think it's important to us to put this on the map of hematopoietic neoplasms. You can see this very long prodrome of evolution involving, in this case, double TET2 mutation in 2 patients 2 decades before they then acquired this very unusual triple MAP kinase mutation within a short space of time. These were difficult to treat patients. It was the pre-inhibitor era. One of the patients we tried to enroll on the basket study, but she was not fit enough to participate in that trial. And I think if we looked back, we would clearly see this was heavily affected by clonal hematopoiesis. We probably, by modern criteria, diagnose them as a form of CMML. For the monocyte count has come down in the criteria of this, and there was some disturbance of the cell count. So, and NGS has become an important factor in diagnosing CMML. So we can see the relationship between this disease. And in fact, the diagnosis of histiocytosis, people focused on this very much and kind of didn't look beyond at the time. We still have some work to do on this. We have to look for the VAF of the mutations in the lesions. We also have a number of patients that have a lower burden of mutation that we are going to interrogate. Method can potentially reveal clonal abnormalities where they're not known chip mutations, so it's agnostic to mutations that might predispose to histiocytosis. And it would be interesting to look back in this category of CMML patients with MAP kinase mutation, which is quite a frequent occurrence, to see if there's any overlap coming from the other direction. And potentially, if we had known these mutations earlier on, you know, they might be sensitive to drugs that would modify the prognosis of the patient. That's something, again, for the future. So I'd like to thank the people that did the work. This is Paul. Many of you have met him before in the lab. A pathologist, Anna Mestri, did some great work digging up the more clinical data. And this is a collaboration with Jyoti Nangalia, who's very famous in the world of myeloproliferative neoplasms for performing this kind of analysis, and her student Kudzai, who did the bioinformatics. And not least, I would like to thank ECD Global Alliance, who funded the sequencing that was involved in this work and the cloning of the cells. And we also received support from these other funders. Thank you for your attention.